Well, welcome everyone to another episode of Conversations with McGill Office for Science and Society, and very special guests. But first, let me introduce you to our usual colleagues. Emily Shore is joining us. Jonathan Jerry is joining us. And we will, a bit later on, talk about monoclonal antibodies, and cloning is when cells multiply. And our very special guest, our favorite communicator when it comes to vaccination, also favored on CNN, is Dr. Paul Offit. He speaks to us from Philadelphia. He's the director of the Vaccination Education Center in Philadelphia at the Children's Hospital. And he's a pediatrician. He's inventor of a vaccine for rotavirus, and is one of the world's leading experts on vaccination, which obviously is a very, very hot topic these days. So Paul, welcome. Yeah. All right, let's get going here. Are we going to have a vaccine by November 3rd?
No. I think that it is likely that we would have a vaccine early next year, assuming things work very well. First of all, there was just an announcement in the last 2 days that these vaccines are not going to be licensed products. Obviously, they're going to be approved through something called emergency use authorization. And there was just an announcement in the last 2 days that they have sort of revised the emergency use authorization protocols so that you now have to wait 2 months after the last dose, and these are all initially 2-dose vaccines, to follow up safety. Well, if that's true, then there's no way this is going to be ready by any time before December, most likely early next year.
Let's very quickly just run through the different types of vaccine that are out there, because I mean, there are some 170 vaccines that are being researched and under consideration, but they fall into relatively few categories, just about 4 different categories. So maybe we can go over those, starting first with the attenuated vaccines, or vaccines that are made from viruses that have been killed.
Right. So that's a strategy with which we have a lot of familiarity. It's the way we make the measles vaccine, the mumps vaccine, the rubella vaccine, the chickenpox vaccine, one of the rotavirus vaccines. You just take the virus and weaken it by sort of serially passaging it in, generally in non-human cells, which introduces a series of genetic changes that makes the virus less capable of reproducing in you, but still capable enough to induce an immune response. Most of those vaccines are actually starting to be phase 2, phase 3 trials, but all outside of the United States and outside of America.
Okay, then we have the vaccines which use just the spike protein of the virus that is directly injected. So how does that one work?
Right. So there, and at least again, we have experience with that kind of strategy. It's how we make the hepatitis B vaccine, the human papillomavirus vaccine, or HPV vaccine, and also it's how we make one of the flu vaccines called Flublok. So there, what you do is you use actually recombinant DNA technology to make that protein. In this case, the SARS-CoV-2 spike protein. You then inoculate the person with that protein, invariably in conjugation with an adjuvant. And usually those are 2-dose vaccines. Novavax actually just started today. I think it's embargoed till 4 o'clock today, so you're hot off the press here. They just started today a phase 3 trial in the United Kingdom, a trial of about 10,000 people. They're planning to make that a 30,000 person trial in the United States.
And the third category is the one that's gotten a lot of publicity, the so-called Oxford vaccine, where they use a virus, a non-human virus that will not replicate in the human body, that has been genetically engineered to produce a protein. So this is quite far ahead, right? That version.
Right. So the so-called replication defective simian adenovirus. So adenoviruses are viruses that can cause disease, a variety of diseases in people, but these are simian adenoviruses. So they're not monkey adenoviruses. So they really don't cause disease in people. And then you make it so that, as you say, you genetically engineer it so it can't reproduce itself. Therefore, it can't cause disease. You also add the gene, that codes for the coronavirus spike protein. So you then inject that virus, and then you make the spike protein and you make antibodies to the spike protein. So that's the AstraZeneca group with the Institute of the Jenner Institute at the University of Oxford in the United Kingdom.
And this is the one that ran into a little trouble a couple of weeks ago.
So that was put on clinical hold in September for a quote unquote case of transverse myelitis, which is inflammation of one segment of the spinal cord. But as we learn now, there wasn't the first clinical hold. This was the second clinical hold. The first one was in the month before that, when there was a case of so-called undiagnosed multiple sclerosis. So see, the problem with these things is, once you put something on clinical hold, once the Data Safety Monitoring Board that's overseeing these vaccines says, you know what, this may be a real safety problem, a real serious safety problem. Let's stop, take a close look at this case, look at other cases, see whether we think this is biologically plausible, look, really get more testing on this person. Once you put it on clinical hold, everyone will know about it. And everybody will assume correctly someone got a vaccine and had a serious event. The problem is, the confidentiality rules dictate that you can't really know all the details of that case, and so it becomes a big transparency issue. And so that's my most asked question: is what's the story with that? And I don't know the story with that because I don't know those cases. I'm just knowing what I read in the newspaper, so it's really hard to comment on it.
And the fourth version is the one that Moderna is working on, is where you inject directly the messenger RNA so that the body will start to produce, it's genetically engineered to produce the protein, the antigen to which the body will react. So which one of these do you think is most likely to succeed?
See, I'm actually on the FDA's Vaccine Advisory Committee, and I actually have to fill out a form that says that I am not allowed to publicly say which among these viruses I prefer. So I won't, but what I'll say is, let's see. The good news is we're doing phase 3 studies, and soon we will know which among these vaccines are safe and effective.
Now, the new Johnson & Johnson one, or the one that they just started the phase 3 trial, is a one-shot vaccine, right?
That's right. That's the first of the one-shots. That's a replication-defective human adenovirus type 26. They just started within the last couple of days a phase 3 trial in the United States. They became the third company to do a phase 3 trial, although the UK AstraZeneca product is still on clinical hold in the United States, even though it's not on clinical hold in the United Kingdom.
Now, one last question about the methodology of how this is going to progress. In the UK, they're talking a lot about viral challenges, that is, having volunteers be voluntarily injected with the virus and then having them be the guinea pigs for vaccine testing, because this, of course, would yield results much more quickly.
Are you signing up, Joe?
Well, I would. I would.
Would.
And they have thousands and thousands in the UK who are willing to do this, because it would be a big contribution to society. I mean, obviously there is some risk associated with it. So I would not be a candidate, because they're only taking young people who, if something goes wrong and they get the disease, they're likely to recover. So Paul, what do you think of these challenge opportunities?
Yeah, I actually, there's a subcommittee that's put together by the NIH. Francis Collins formed a group called at the NIH called the Active Group, which is a group designed to accelerate and facilitate the development of a vaccine. We actually had a subcommittee looking at this, looking at whether or not we wanted to do human challenge trials in the United States. The committee basically decided not to, and the reason was twofold. One is that under natural conditions, when you're out there getting infected with SARS-CoV-2, you will be exposed to different quantities of the virus. And there is an inoculum effect, meaning that generally, as a general rule, the lower the inoculum of virus that you get, the more mild your symptoms. The higher the inoculum you get, the more severe your symptoms, more likely to be moderate to severe. You can't really mimic that in human challenge trials. I mean, generally they will pick a dose to challenge them, and generally it will be a dose that causes sort of mild disease. You're not looking to send people to the hospital or to the morgue when you have a virus that can kill you and doesn't have a rescue drug. And also, as you know, you can't really do those studies in people who are older, which is clearly an important target group, because again, those are people who are much more likely to die, and there's not a rescue drug. So that's not something we're doing in this country.
Interesting that you brought up the inoculum, the dose of the virions that make our, because there was just this paper in the New England Journal that came out, where it's a very interesting idea, where they talk about masking. And of course, as a lot of issues with masking, especially in the US, where, last week they had a mask burning party. I mean, unbelievable. But anyway, the contention here is that while a mask obviously does not protect you totally from viral particles entering, it allows a small amount through, and that may just be enough to trigger antibody reaction in the body. So, or you may get a slight version of the disease, but not much. So, what do you think of this? That this is sort of an added benefit of the mask, that
a very interesting
actually induce antibody formation.
It's an interesting hypothesis. I mean, there certainly is an inoculum effect. It's the old it's the second child in the house who gets chickenpox who usually gets the more severe disease because they're in much closer contact with that first child who gets it. It's an interesting hypothesis. I'm not sure exactly how you would test that hypothesis. Maybe you could put like masks on ferrets and then send them to some maskless bikers conference in South Dakota. You could do it that way. Or maybe,
but they have put masks on Syrian hamsters, right?
I was going to say Syrian hamsters. I should have said that. Right?
Okay.
But
I mean,
when we said, and maybe it's a silly question, but there was we haven't spoken with Paul since there was reports of the person getting reinfected twice, right? Or maybe I think there was even 3 people. But when they looked into that, they were talking about because that person got it the first time and it was so little, they had no symptoms. He was almost totally asymptomatic, so he didn't really build up the antibodies for that and everything. So it's an interesting, I guess, like Paul said, hypothesis to have the masks and just allow a little antibody build up. But if that even were to happen, it would not even be enough to avoid getting reinfected for real a second time, I imagine. Right?
It's when you make a vaccine, what you want to find that natural infection protects against disease. I mean, if natural infection does not protect against disease, you're in trouble. And there's plenty of examples of that. You get group A strep, strep throats over and over again. You get gonorrhea one gets gonorrhea over and over again. Because when you see that, you're going to have trouble making a vaccine. That's why we don't have vaccines for those 2 things. When we developed the rotavirus vaccine, the best news was children who had rotavirus infection in the first year of life, when they were then subsequently naturally infected, when they're second, third, fourth year of life, they had much more mild disease. And that's what you want to do. That's what you want to see. That's what you think a vaccine can do. And I think that's what SARS-CoV-2 does. I mean, if you look at people who are infected sort of with moderate to severe disease, and then they get reinfected, and they can get reinfected, usually they have more mild disease. That's the usual story. So I take great heart in that. And that's what I expect from a vaccine. There was an article just in the New York Times, which has become the New England Journal of Medicine, by the way, since that's where you get most of your medical information.
You mean the failing New York Times?
Failing New York Times, yes. There was an article by Peter Doshi and Eric Topol where they made a statement that I think wasn't true, that they didn't like it that the current clinical trials are really looking for prevention of mild to sort of moderate disease, but don't really have severe disease as part of their protocol. But see, I think if you can prevent mild disease or asymptomatic infection, that's much harder than preventing against moderate to severe disease. I mean, our vaccine, rotavirus vaccine, prevents moderate to severe disease. It really doesn't prevent as well asymptomatic infection or mild asymptomatic infection, which is fine. You're just trying to keep people out of the hospital and keep them from dying. But so I was surprised that was published, but by 2 people, frankly, that aren't vaccinologists, which is part of the problem.
Let me bring Jonathan in here, because he's been looking at the situation in Sweden. And, I mean, this has been controversial. Sweden has been less rigorous than many other countries, although it is not true that they had no recommendations. I mean, they had all kinds of recommendations, and Swedes were much more likely to follow the government directives. So, Jonathan, what about the Sweden story? Are they achieving herd immunity?
Well, not according to serological surveys that were published in April. So, I mean, it's been a little while, but serological surveys are when you, hopefully randomly, but sometimes out of a sample of convenience, you take blood draws from people and you check for antibodies against the new coronavirus, and then you extrapolate that data to see what percentage of your population has these antibodies and thus is probably protected against the coronavirus. And the number that is being floated around to achieve this community immunity is about 70%. 70% of your population would need to have these antibodies. And in the case of Sweden, I mean, in Stockholm, they found about 17%, 17. In Malmö, which is in the south of Sweden, it was much lower than that, it was something like 6 or 9%. So they're not anywhere near that if we trust these serological surveys done in April. But as you mentioned, I mean, there's been a lot of confusion about what Sweden has and hasn't done. I mean, there was no mandated lockdown, in large part because their law does not allow for the quarantining of entire cities. But there were recommendations very similar to the recommendations that we had here in Quebec, that were voluntary and that were adapted from the WHO by their public health agency. So it's very hard to sort of adjudicate if their approach was good or bad. There are certain things where I was a little iffy about how they were going about things. But their main idea was, let's do something that we can keep up for a long period of time. Whereas, we had the lockdown and then we opened up again, and now we're sort of slowly going back indoors. So again, time will tell, which method was the best, and it might vary from place to place, from culture to culture.
I remember being in Stockholm a few years ago, of course, long before COVID, and surprisingly, people at bus stops were lining up so that they were distanced from each other, just out of courtesy, because you won't be on top of someone. Here, of course, the bus stops and everyone rushes at it. So I think that the Swedish culture is much more prone to being distant from people.
To a point, I mean, I was reading about this, and it's also important not to paint with too broad of a brush this idea of Swedish culture, because there's a lot of variation within the country, and culture can change as well. I mean, it's based on traditions, it's based on the evolving social landscape. So so this idea that Swedes are very compliant, it might be a little reductive.
Paul, what do you think the chance of another total lockdown?
In the United States, zero.
Yeah.
I think our current administration's plan is to just get out there, and if that means that you're more likely to suffer or be hospitalized or die, so be it. I mean, this is an administration that makes fun of people that wear masks, that makes fun of people that social distance. So this key component for eliminating this virus, which is hygienic measures, is pretty much thrown out by this president. But one thing about natural immunity I want to say is that name the virus that has been eliminated from natural immunity induced by, from infection. There is none. It's never happened. I mean, if you have to pick the perfect virus to induce herd immunity, it would be measles. I mean, I was infected with measles as a child. That means that I am protected against measles for the rest of my life completely, meaning I will not develop even asymptomatic infection. So it's complete long-lived immunity. Yet still, every year in the United States, about 2 to 3 million people would get measles, about 50,000 would be hospitalized, and about 500 would die. So there's no such thing as that. I don't think it's possible. We have, there's 4 strains of human coronavirus that circulate every year in the United States, and they account for 15 to 20% of the respiratory infections we see come into our hospital every winter, and that's never changed. I think this Swedish notion that you could ever induce population immunity with natural infection was always wrong.
Testing, of course, is in the news all the time. And as we hear virtually every day, the US does more testing and better testing than anyone in the world. I just saw that Abbott has come out with a new antigen test that you don't have to send into the lab. 15 minutes and you see the color change. The only, I think, the proviso here is that it has to be done within the first 7 days after symptoms have set in. So where do you think generally testing stands, and where is it going?
Yeah, I'm going to say something that's somewhat contrary to public health position on this. There's a lot of asymptomatic and mildly symptomatic people who shed virus, and so we test them haphazardly and sporadically. We're not capturing most of those people. So I think the most sensible thing to do is to assume that everybody you come in contact with is asymptomatically infected and shedding, who you don't know. And just, and so when you're out there and you're close to somebody, just wear a mask and do the best you can to stay away. Or said another way, I don't think we're going to test our way out of this pandemic. But I know that's not the public health view. The public health view is test, do contact tracing, quarantine. It's just done in such a sporadic, haphazard way that I just don't think it's had the kind of effect we would hope it had. Or maybe if we did it the way South Korea has done it, then we would have had a much better chance of doing this. But we never had that kind of federal leadership. So it's just been
Why? Explain, just for those, and just remind people about South Korea.
Right. So South Korea was able to really get on top of this infection, as has Japan, Singapore, Germany even. I mean, have done much better than we have. I mean, the United States has roughly 4% of the world's population and 25% of the deaths, for which Donald Trump has given himself an A. I mean, what
A plus.
No, A plus only if there's a vaccine. He said he gets an A, and when there's a vaccine, he gets the A plus. Actually, I had to give a webinar yesterday, and I said, okay, we're going to talk about the only thing that's keeping Donald Trump from getting an A plus vaccines. I think, it's what gets you a B in this, 50% of the world's deaths.
What about the test that, I think that was what the NBA was using. And I also heard here in Montreal, even schools are maybe going to start using it, the saliva one, right?
Certainly it's faster and easier. And so that there's a tremendous advantage to that. I mean, so that sort of deep nasal swab thing was like a brain biopsy. I mean, that was too painful.
So is that not being used anymore?
It's being used. No, it's being used less. I know in our hospital, we don't use that. We do more the interior sort of areas.
Okay.
I think Taiwan also has done very well, right?
Right.
And I think they're also because they already had a culture of using masks. You often see people walking around with masks on the street long before COVID, when they just thought they had a cold.
Yeah,
anyway,
one thing I think is interesting is it'll be interesting to see what happens with flu this year. I mean, I'm sure there will be a dramatic drop in the incidence of flu in the United States, as there was in Australia and in the Southern Hemisphere, because now we're wearing masks and social distancing. So, last year flu caused 780,000 hospitalizations and 60 deaths in the United States. We could dramatically reduce this by wearing masks, by changing our culture. I don't think that's going to happen, but.
And if people get the flu shot.
Yeah, that's a good idea too.
Yes, I wanted to ask you about the flu and the flu shot and everything. Make sure people get it.
Absolutely.
Yeah.
No, absolutely. And of course, the nonsense keeps coming up, and even so-called experts are weighing in. And Jonathan has been looking at a character physician who has been spreading information that is, let us say, highly questionable. So, Jonathan, you want to just pipe in with that?
Sure. I'm wondering, Paul, have you heard of Dr. Andrew Kaufman?
I haven't. I know there's a comedian, Andy Kaufman, right?
Yeah, I know, but this Andy Kaufman is no joke. So Andrew Kaufman, I became aware of him at the very beginning of the pandemic because there was a video that got very, very popular that was making the rounds, where this guy that I didn't know, this Dr. Andrew Kaufman, a medical doctor, was arguing that there was no coronavirus. But he was doing it in a way that was very calm, very, he sounded very knowledgeable. He had all these slides, and he was claiming that it was all exosomes. Now, for people who are watching who don't know, exosomes are very real. So we have our cells, and sometimes we have these little bubbles that bud off of the cell, and they can act as little transport vehicles. And sometimes they carry genetic material like DNA and RNA. And so he did this 45-minute presentation claiming that there was no coronavirus, it was all exosomes. And that video spread around and around and around. And since then, he has kept going on, and he does these hour-and-a-half live streams where he doles out medical information, where people write in with their symptoms, and he gives them advice. And turns out that he's a psychiatrist who still has an active medical license in the state of New York. But he is appearing to be turning his back to medicine and embracing a form of naturopathy. And I spent, a few days just watching hours and hours of his videos for an article that will be published on Saturday. And some of the things that he says are absolutely, insane, in my opinion. He claims that appendicitis, for example, is no big deal. It's just constipation, and you can relieve it with an enema. But of course, doctors are paid more for a surgery than they are for an enema. He claims that herpes, there's no herpes virus. He's a virus denier. I mean, he believes that viruses exist, but none of them cause infectious diseases. And so herpes is just the collagen deficiency. He tells a lot of his viewers to start drinking bone broth because they all have collagen deficiencies. But the thing is that he has the beard. He's probably in his 40s or something. He's got a bit of white in the beard. He's bald. He's got the glasses. He speaks in a very, very even-tempered keel. He sounds very, very knowledgeable, and he reaches hundreds of thousands of people. And he was a part-time doctor still recently, and he lost his job because he was refusing to wear a mask. And now he's leading a group of people in Syracuse, New York, where he lives, in anti-mask demonstrations in like the supermarket. They just barge in 40 of them without masks to make a point that there is no pandemic. And of course, he espoused a lot of conspiracist ideation that this is all a ploy by the government to have soldiers force you to get a vaccine and to force you to get government handouts, et cetera, et cetera, et cetera. I mean, it was really something to hear him say some of these things, because if you listen to like a 20-minute stretch, he might sound, I mean, obviously he thinks that there are toxins everywhere, but it's nothing unusual that you haven't heard before from naturopaths. But once in a while he says something that really made me drop my jaw.
An hour and a half is a long time to do one of those videos. He's probably right. 20 minutes, okay, fine. An hour and a half.
Do you know why he does this? Is because he charges for virtual consults. He charges about $750 American dollars, and so these free live streams are a way for him, in my opinion, to gain clients, because he says, well, given what you're telling me, it could be this, could be that. Go to my website and file for a consult, and I'll give you more information. So, you do the free live stream, you give some information, then you say, well, it's a bit complicated. Why don't you hit back at me? And the premium package is $1,750 US.
Well, for more information,
I was going to say, maybe
our website, because Jonathan's takedown of this critter is going to be there for this Saturday's newsletter, right? And it's much more detailed, and it's a real eye-opener.
Yeah.
All right, let's leave the quacks aside for a moment and get back to something that may actually work. Vitamin D and zinc. So Paul, I mean, there's a lot of talk about this now. I mean, zinc, of course, is certainly important in immune function. Nobody debates that. But what about the wisdom of taking a small dose of zinc supplement to try to reduce the severity of the disease should it strike? And
vitamin D, I've heard a lot.
Yes, we'll talk about that too.
Mm-hmm.
Yeah, I guess, so anything's testable. I think if there's certainly enough SARS-CoV-2 infections out there that one can test this, that's all I would ask. I mean, vitamin D has been, put forward to do a lot of things, and observational studies and ecological studies have sort of supported its use. But then when you do randomized control studies, as a general rule, vitamin D, the bloom comes off the rose. Zinc was also studied, and the data are not quite as clear, but I guess study it. That's all I would say.
Yeah, there are a couple of studies that just came out actually yesterday, which is one of them is about vitamin D. And it was a proper interventional study where they used the metabolite of vitamin D3, which is the actual hormone. And they showed that they were able to, once the patient was hospitalized, they were able to reduce the chance that they would have to go into the ICU with vitamin D. So, I mean, it's interesting enough to trigger further studies. It's not something that can be dismissed. Also with zinc, there's a video that's going around, I know, because so many people have sent it to me, by Dr. Ritchie, who's, I don't know if you've seen this, he's a rabbi, Orthodox guy, but he's certainly a legitimate doctor. And at one time, he had a couple of papers published in New England Journal about 30 years ago about vitamin E in premature babies. So he's certainly not in the quack category. And he talks very calmly, and he says that everyone should be taking a zinc supplement, 30 to 40 milligrams. But he sounds very authoritative. And he doesn't quote any references in this video, but he very smooth talker,
and yeah, he's very calm. The same type of calm probably as that Kaufman guy.
Yeah. So I mean, the zinc and the vitamin D3 are certainly interesting to follow. Now the hydroxychloroquine, I mean, you would think that this would have been buried already. But now, interesting enough, it's being resuscitated with the zinc argument, because apparently hydroxychloroquine acts as an ionophore, that it transports zinc across the membrane. So now, they're doing studies that, yeah, maybe it doesn't do anything by itself, but together with zinc, it does.
What about with vitamin D? Let's try all the combinations.
Yeah. And Paul, what about that group of doctors? They call themselves the doctors at the frontier or something, who stood there in front of Congress, in the front of the Capitol in Washington, and were singing the praises of hydroxychloroquine as a cure. The lead doctor was one who said that people were also doing experiments with drugs to keep people from being religious.
Yeah, I think that was also the same person who said that the women had, for example, that had uterine problems. It happened because they had dreamt that they had slept with the devil.
Yes.
I think this is, you can't make this stuff up.
No.
Well, Dr. Kaufman thinks there's a good reason to believe that demons might be involved in mental illness, and he's a psychiatrist, so they'd be great bedfellows. But I had a question for Paul regarding a particular sort of vaccine debacle, which is rearing its head now as because of Operation Warp Speed and all of that. I'm hearing from a lot of anti-vaxxers, people who are vaccine hesitant, about this 1976 swine flu vaccine debacle that happened in the United States. And so I was wondering if you knew about this, if you knew what had happened, and if there are lessons that we can learn from what happened back then.
Yeah, what was the debacle?
Right. So there was an outbreak of influenza in Fort Dix, New Jersey, in the barracks in Fort Dix, New Jersey, military barracks, which was, when isolated, was a strain that had the potential to be a pandemic strain, which is to say a strain that was novel, that others hadn't seen, most people had never seen before. With that, there was a decision to launch a program in the United States by Gerald Ford to get everybody immunized. As it turns out, it never was a pandemic. And I think that what they should have done is waited a little longer. When you're in a barrack situation, you're much closer to people. There's like 60 people per barracks. They're all very close together. It's not a natural situation. They really should have waited just a moment to see whether there was any evidence that it spread outside of that Fort Dix, New Jersey Army barracks, which it really never did. But Ford was convinced that by his medical experts, this is what happens when you listen to your medical experts, this was going to be a problem. And so we administered about 40 million doses roughly of the swine flu vaccine in 1976. I was one of those recipients, actually. It was given by a jet gun. I remember getting that vaccine. Oh. The problem was that there was an untoward side effect, which was Guillain-Barré syndrome, this so-called ascending paralysis that can be severe and occasionally fatal, that occurred in roughly 1 per 100,000 people. So it's extremely rare, but it probably was real. And occasionally with other flu vaccines, you'll see every few years will pop up the notion that Guillain-Barré syndrome may be a consequence in roughly 1 per million vaccinees. It should be pointed out that natural influenza virus also causes Guillain-Barré syndrome at a rate roughly 17 times greater than that caused by the vaccine, which therefore means that the vaccine at some level prevents Guillain-Barré syndrome. But that's what happened then. And he was heavily criticized for that. And occasionally I'm asked the question, didn't Ford already know about that? Well, he couldn't have possibly known about it. It occurred in 1 per 100,000 people. There was no way to know about that unless the vaccine was first given to millions of people, which is what happened.
Thank you.
Okay, let's get back to the vaccine and when it's likely to come out. I think the safety factor can be addressed relatively easily, right? I mean, the phase 1 and phase 2 trials will give you a pretty good idea of safety, right? Because if there's any serious problem, it would crop up in the first few months, right?
I think you can rule out a common side effect problem, serious side effect problem in phase 1, which usually 20 to 100 people. I think in phase 2, which is hundreds of people, you can rule out a common to mildly uncommon side effect problem. Then when you get to phase 3, where you're in tens of thousands of people, I think you can rule out a relatively uncommon side effect. You're not going to rule out rare side effect rare serious side effect problems
Right.
till post-licensure. But yeah.
The issue, the way I see it, is more of the efficacy, because that you're not really going to know until years have passed, right? So, I mean, everyone is jumping on the vaccine bandwagon and thinking that, maybe we'll have a vaccine next year and that this is then the solution to the problem. Well, we'll have a vaccine, or more than one vaccine probably, that are deployed. But how long will it be before we will really know efficacy?
Right. I think you can know efficacy in the short run. I think when vaccines are licensed and recommended, you usually have efficacy data for a certain period of time. Here, it's going to be a relatively short period of time. It'll be months. I mean, when the rotavirus vaccine, you knew that efficacy based on our trials was about persisted for about 4 years. With the HPV vaccine at the time of licensure, you knew it persisted for about 7 years. So here it's going to be months. So you're right. You're not going to learn how long this effectiveness lasts, or to what extent it wanes, until after it's licensed. And that's going to be a challenge. What worries me the most in this, actually, Joe, is that the vaccines are being sold as like this magic dust that we're about to sort of sprinkle over the United States and make this pandemic go away. I mean, it's going to take two things to make this pandemic go away: hygienic measures and a vaccine, and one is not going to replace the other. I think both have to be going on at the same time. I was on service last week. Our hospital has a number of children in it that are infected with COVID-19. If you gave me a choice to do one of two things, I could either wear a mask and do the best I could to social distance, or get a vaccine. I would take the mask every time. I mean, the mask does a pretty good job at preventing small droplets from coming into my nose or mouth. Let's say the vaccine's 75% effective, which I think everybody would be ecstatic with, right? Mm-hmm. 75% effective against moderate to severe disease means 1 out of every 4 people that gets it could still get moderate to severe disease. And it probably will be less effective against mild or asymptomatic disease, which you're still shedding. So what worries me, what keeps me up at night, is the notion that people will get a vaccine and think, great, I'm good. I don't need to wear the mask. I don't need the social distance. But really, you still need to wear the mask to protect you and others. The vaccine is not going to be 100% effective.
But can I
Not
ask?
only that, you have 30% of the population who say that they will not take the vaccine no matter what.
Yeah, I would say let's wait till the vaccine comes out and then is given to hundreds of thousands, then millions of people, and it's still safe, hopefully. And then you'll have more data, so you can at least say, here's the characteristics of this vaccine. Right now, when you ask people whether they get a vaccine, you're asking them whether they get a theoretical vaccine. I mean, if you ask me that question, I would say no, not till I see the data. And so let's see what happens with the data.
Can I ask a question? I mean, there's a lot of talk saying, like, when there's a vaccine, of course, there's going to be multiple producers of the vaccine and everything. So for those of us who are not scientists and have never really paid attention to like vaccine development before in a pandemic anyway, what vaccines, I imagine do we take now on a regular basis every year, or children or whatever, that there are multiple makers of those vaccines? Like when I heard, there's going to be a lot of different vaccines on the market, I guess. Is that a normal thing for there to be many different vaccines and many different producers? And is there one that's better than the other?
So most vaccines, like the hepatitis B vaccine, has a couple, 3. Now there's 3 manufacturers of the hepatitis B vaccine. The human papillomavirus vaccine in the United States has 1 manufacturer. The shingles vaccine had 2, now it's 1. So that's usually close, usually a couple. The one that has the most manufacturers is influenza vaccine.
And are they all the same vaccine that they're producing?
No, so with influenza, for example, there's the high dose vaccine. There's the squalene adjuvanted vaccine. There's the traditional just quadrivalent vaccine made in eggs. There's the mammalian cell quadrivalent vaccine, which isn't made in eggs, made in mammalian cells. There's like at least, then there's flu block, which is a recombinant DNA vaccine. There's at least 5 different strategies that are used to make the flu vaccine.
Okay, so it's not like abnormal that there could be, even when a vaccine in 5 years, God willing, is going to be good and everything, there might be multiple vaccines. And that's normal.
That's likely. And then I think what we'll find out is that there'll probably be, it's what happened. Sometimes vaccines replace one another. I mean, the Zostavax shingles vaccine was replaced by the Shingrix vaccine. It was a better vaccine, much better. We may find that 2 years from now, assuming this virus is still with us, that and I suspect it probably will be at some level, that there's a better vaccine. So then the question will become, can you get this other vaccine? Is there compatibility or incompatibilities between those 2 vaccines? That'll be the question.
There's also going to be the production issue, right? And you're talking about potentially billions of people who will need the vaccine. So even once there is a working vaccine, producing it on that scale is going to be a challenge. And then,
you're right. And you know what, I mean, how much more information do we need that what happens in other countries can affect our countries? You're only as strong as the country which has the biggest outbreak. So you need to, we have an obligation to make sure that, that everyone gets this vaccine.
Now, what are treatments? Eli Lilly, new treatment with its monoclonal antibody, which looks pretty good on paper and the preliminary experiments, but there, I mean, I think the major issue is production and cost. I mean, the cost of monoclonal antibodies is exorbitant. You know, I mean, anyone who's, you know, had to use these for arthritis or cancer or whatever so far, I mean, these are hundreds of dollars per treatment. So even if we have something like that that works effectively, it's going to be such a small fragment of the population that is going to be able to use that.
Yeah, and the thing with antibodies, which is to say antibodies that neutralize virus infectivity, usually viruses replicate at the beginning of illness. I mean, that's when, you know, you're infected, the virus starts to reproduce itself 10 times, 100 times, 1,000 times, and it, you know, then viral replication comes up here. Then you start to make an immune response. You know, the immune response is now countering the virus. The immune response is what causes your symptoms. And then as the symptoms worsen, you know, the amount of viral replication actually decreases. So if monoclonal antibodies or convalescent plasma are going to work, you really need to give them very early on in infection. And that's why that big Mayo Clinic study of 35,000 people didn't really show that convalescent plasma worked. I mean, I'll give you another example. If you're exposed to measles or hepatitis A or chickenpox or rabies, you get immunoglobulins, right? You get rabies-specific immunoglobulin or varicella zoster-specific immunoglobulin or measles or hepatitis A virus immunoglobulins, and that works really well in the first couple of days. But if you look at chickenpox, for example, if you're exposed to chickenpox, there is a chickenpox-specific immune globulin that you can get that works very well in the first 2 days. It doesn't work that well after day 3. And you're still within the incubation period of that disease, and it still doesn't work very well. So I think it never made sense to me that convalescent plasma would work, actually. And then, you know, obviously in this country, you know, the FDA was sort of bullied into releasing it, even though it was something the NIH was not happy about at all. And both Francis Collins and Tony Fauci said we shouldn't do this. Although I was on when they had that press conference and Donald Trump said, this is a breakthrough therapy, this is a major breakthrough. I was on an NIH conference call that afternoon, and one of the NIH researchers said, yeah, it's a breakthrough. Maybe it'll win the Nobel Prize in 1890, right, when we first started to use convalescent plasma.
There was just a very large study I saw from India on convalescent plasma, which showed no effect.
Right.
It was very significant study, and yeah, so I don't think convalescent plasma is going to be the breakthrough. Of course, he's very fond of breakthroughs and game-changing things, but so far none of those have come true. So Paul, let me ask you this: since this whole thing started, and with the expertise that you have in this, what changes have you made in your own life?
Well, I mean, my life is
living at the beach.
Here I am at a beach house, right? I mean, I am really careful when I'm over 65. I wear a mask whenever I'm with people. I do my best to socially distance. I really don't like to eat inside at restaurants, although my wife and daughter and her boyfriend do. So I will not eat at a restaurant where there is another table within 6 feet of my table. And, you know, my life is dramatically changed. It's you know, I'm scared of this virus. I am. And when I'm in the hospital, it's interesting, when I'm in the hospital, I'm not as scared. I mean, in the hospital, you know,
I hear that from my doctor friends too.
Because, you know, you have to, by rule in our hospital, not only do you have to wear, obviously, a mask, but you have to wear eye gear, you know, just goggles to also protect the mucosal surface of your eyes. And everybody does. And everybody's very careful about that. So, and, you know, I think there's only been 2 hospital workers, despite the fact we see a lot of COVID in our hospital, and it's a children's hospital. Only 2 workers have ever got it in the hospital. So, and, you know, we have a huge staff. We have a 700-bed hospital.
Are you treating COVID patients? Like, have you seen them? Children?
Yep. COVID and also MISC, which is sort of the so-called multi-system inflammatory syndrome of children, which is probably a post-infectious phenomenon. Yeah, we see that. I think pretty much anybody who comes into our emergency department who has a fever is considered to have COVID-19 until proven otherwise.
Which mask do you wear in the hospital?
I just wear a surgical mask. Just the, the rectangular surgical mask. If I'm going to walk into a room of somebody with COVID-19, which I really don't want to do, to be honest with you, I mean, let the fellows do that and then tell me what they saw. Then I'll wear an N95 respirator.
And around the home, like when you are with people, you just wear the surgical mask?
Around the home, I mean, it's I don't wear the mask here because everybody, I know them here. But when people come over, I'm not crazy about it. When, because I've been filmed a couple times and I have people that do the filming stay in the back, and they have to wear masks. And obviously anybody who walks in who has to repair something has to wear a mask, and I stay away. Yeah, it affects our lives. I think what's the heinousness of this virus is anybody who you see, you think could be shedding this virus because of asymptomatic spread. With smallpox, for example, I mean, there's no such thing as asymptomatic smallpox. If you have smallpox, not only do you have these blisters, you have them on your face. And people are contagious because the lesions on the inside of your mouth aerosolize. So it's really large droplet spread, not small droplet spread. So you have to be fairly close. But here you don't know.
No, it's best to assume that really, like you said, that everyone kind of does have it. Or when the mask comes off, we talked a lot about mask handling, that when the mask comes off in your house, pretend it has virus on it and then treat it as such. Wash it, laundry, whatever, I think.
Anyway, I see it's 2:45, and Paul, I know that you have to go because CNN wants you.
Really, we are losing you to see.
But they'll give you the usual 1 minute.
That's right. Here I get some time. Right there, you get 1 minute. And what amazes me about CNN is I see all these other infectious disease people on, and they look great. There's they're well lit and they have the microphone and they're just infectious disease people like me. How did they do this? I always look like one of the bad guys on Saw 3. And they look great.
Because they're paid actors. That's what Alex Jones would say.
Right. You look good, and we schmooze. So we gave you more time.
What do they want from you this time?
They want me to comment on Trump's comment yesterday that he was going to bypass the FDA.
Okay,
we'll be listening. Tune in right now.
Yeah.
Benefits of working from home. Yeah.
So thanks a lot, Paul, for joining us. And
thank you so much again.
All the updates, and
we'll have you back.
I think what I'm going to do now is I'm going to wait to see when Donald Trump makes Andrew Kaufman his chief science advisor.
Well,
give it a few months.
Yeah.
Apparently, Deborah Briggs already is having trouble there, right? She says that she can't live with this, and Fauci, of course, has been shunted aside. And Trump says that he likes him, but somebody disagrees with him. And he has no right to disagree. He knows nothing. He knows nothing.
But he has the genes of the researchers, right? Because wasn't somebody in his family that were very bright.
His uncle. He's got his father's genes too, and unfortunately, there are some questionable genes there. So
yeah.
All right.
Thank you, Paul.
So, thanks, Paul, and thanks, Jonathan. Thanks, Emily.
Oh, we'll see you in 2 weeks back with Dr. Debbie Schwartz, who will be giving some Montreal update because Quebec and Montreal is not doing wonderful right now. So we will see what happens with Debbie.
Yes. Dr. Debbie is not quite in Paul's class, but she
knows her stuff.
What she's talking about.
Yeah.
She's also got some good genes.
Exactly.
Okay. Thanks, everyone.
Bye.
Bye.
Bye.